Welcome to the AJP-Heart and Circulatory Physiology April edition of the video table of contents, or from the editor's desk. The past few months have been surreal, if not difficult, for everyone. We hope you will find some distraction in this month's issue. Also in this issue, we highlight the best AJP Heart and Circulatory papers awards that we usually present at the cardiovascular section dinner at the experimental biology meeting. We have some really interesting and relevant articles this month. There's something here for everyone. I'm going to highlight just a few and then say a few words to our readers, reviewers, and authors. Now let's take a look inside. In a review article by Zhao et al., the role of and mechanisms by which estrogen deficiency leads to cardiovascular diseases are discussed. Recent research findings provide evidence that estrogen plays a pivotal role in the regulation of calcium homeostasis and therefore fine-tunes normal cardiomyocyte contraction and relaxation processes. Disruption of calcium homeostasis is closely associated with the pathological mechanisms of cardiovascular diseases. Thus, the review maps out and summarizes the effects and mechanisms of estrogen on calcium-handling proteins in cardiomyocytes. The second review article, Dominic and colleagues review the literature related to how HIV results in cardiovascular disease. They review cardiovascular disease onset in people living with human immunodeficiency virus and focus on 1. The spectrum of cardiovascular complications that typically manifest in such people, and 2. Underlying mechanisms that are implicated in this process. Here, the contribution of such factors and modulators and underlying mechanisms are considered in a holistic and integrative manner to generate a unifying hypothesis that includes identification of the core pathways mediating cardiovascular disease onset. The review focuses on sub-Saharan Africa, as there are relatively high numbers of people living with HIV residing within this region. Miller et al. Determined whether DNA damage-free iPSCs, or DDF-iPSCs, can be selected using stabilization of p53, a transcription factor that promotes apoptosis in DNA-damaged cells, and differentiated them into functionally competent DDF cardiomyocytes, or DDF-CMs. p53 was activated using Nuclein 3A in iPSCs to selectively kill the DNA-damaged cells, and the stable DDF cells were cultured further and differentiated into cardiomyocytes. They observed a significant decrease in the expression of reactive oxygen species and DNA damage in DDF iPSCs compared with control iPSCs. The differentiated DDF cardiomyocytes had a compact beating frequency between 40 and 60 beats per minute, accompanied by increased cell surface area. The results indicate that DDF iPSCs could be selected through p53 stabilization using a small molecule inhibitor and differentiated into ventricular DDF cardiomyocytes with fine-tuned molecular signatures. Kang et al. Hypothesized that sulforaphane reduced right ventricular fibrosis and mass by an antioxidant and anti-inflammatory effect in a model of pulmonary hypertension. Pulmonary hypertension was produced by administration of the VEGF antibody SU-5416 and chronic hypoxia. SU plus hypoxia induced progressive right ventricular but not left ventricular diastolic and systolic dysfunction and right ventricular and pulmonary remodeling, fibrosis, and inflammation. Sulforaphane prevented SU plus hypoxia-induced right ventricular dysfunction and remodeling, reduced RV inflammation and fibrosis, and upregulated NRF2 expression and its downstream gene NQO1. Sulforaphane also reduced pulmonary hypertension-induced vascular remodeling, inflammation, and fibrosis. Thus, pulmonary hypertension-induced right ventricular and pulmonary dysfunction, inflammation, and fibrosis can be attenuated or prevented by sulforaphane, supporting the rationale for further studies to investigate sulforaphane and the role of NRF2 and the NLRP3 pathways in preclinical and clinical pulmonary arterial hypertension. As many of you know, the journal selects 3 Best Paper Awards using a variety of citation metrics. These awards are usually presented to authors at the annual cardiovascular section dinner at the experimental biology meeting. Because EB was canceled due to the COVID-19 pandemic, we would like to congratulate our winners and highlight these papers here. The best original research paper this year, entitled Protective Effects of the Mechanistic Target of Rapamycin Against Excess Iron and Ferroptosis in Cardiomyocyte, goes to Drs. Baba, Higa, Shimada, Horiuchi, Suhara, Kabayashi, Wu, Aoji, Marth, Kitoka, and Matsui from the John Burns School of Medicine at the University of Hawaii, Manoa. The next award for Best Review Paper, entitled Flaxseed, Its Bioactive Components and Their Cardiovascular Benefits, goes to Drs. Preek, Nedekan, and Pierce from the University of Manitoba. Congratulations. Finally, the Article Impact Award goes to a paper entitled Doxorubicin Triggers Splenic Contraction and Irreversible Dysregulation of COX and LOX that alters the inflammation resolution program in the myocardium. This paper for 2020 goes to Drs. Jadapali, Wright, Kane, Sherwani, Sonkar, Yusuf, and Halade from the University of Alabama, Birmingham. Congratulations. Thank you for watching the April AJP-Heart and Circulatory Physiology video table of contents. The last several months have been trying on everyone in every industry. Education and research have been markedly affected by the COVID-19 pandemic. Laboratories have cut back capacity due to university and industry personnel being required to work from home. Important conferences have been canceled, and the celebration of our accomplishments have been cut way back. We at AJP Heart and Circulatory Physiology understand the potential difficulty you might have in submission of new or revised manuscripts within specific deadlines. The editorial staff is here to evaluate every situation on an individual basis. If you need additional time for submissions or have any other concerns due to the changes in your research environment, please contact us and we'll do everything possible to assist you. In this regard, this may be an ideal time for you to write up the results of studies that have been sitting around for a while. Consider sending us rapid reports that make a single point and can be reviewed and published quickly. If any of you are doing work related to the COVID-19 and cardiovascular function, please get in touch with me so we can assess its suitability for rapid publication. Please stay safe, and we'll see you next month. I hope under better circumstances. And in the meantime, we value your input, and please, as always, don't forget to send us your best work now.